Alpha-defensins are a family of mammalian defensin peptides. Defensins are 2 to 6 kDa. Cationic, microbicidal peptides active against many gram-negative and gram-positive bacteria, fungi, and enveloped viruses, containing 3 pairs of intramolecular disulfide bonds. On the basis of their size and pattern of disulfide bonding, mammalian defensins are classified into alpha, beta and theta categories. Alpha-defensins, which have been identified in humans, monkeys and several rodent species, are particularly abundant in neutrophils, certain macrophage populations and Paneth cells of the small intestine. Defensins are produced constitutively and/or in response to microbial products or proinflammatory cytokines. Some defensins are also called corticostatins, CS, because they inhibit corticotropin-stimulated corticosteroid production. The mechanism, S, by which microorganisms are killed and/or inactivated by defensins is not understood completely. However, it is generally believed that killing is a consequence of disruption of the microbial membrane. The polar topology of defensins, with spatially separated charged and hydrophobic regions, allows them to insert themselves into the phospholipid membranes so that the hydrophobic regions are buried within the lipid membrane interior and the charged, mostly cationic, regions interact with anionic phospholipid head groups and water. Subsequently, some defensins can aggregate to form channel-like pores. Others might bind to and cover the microbial membrane in a carpet-like manner. The net outcome is the disruption of membrane integrity and function, which ultimately leads to the lysis of microorganisms. Some defensins are synthesized as propeptides which may be relevant to this process. Alpha-defensins of the mouse bowel were historically called cryptins when first discovered. Human defensins. Initially, human alpha-defensin peptides were isolated from the neutrophils and are thus called human neutrophil peptides. Human neutrophil peptides are also known as alpha-defensins. Sequences of major human alpha-defensins, HNP1, HNP2 and HNP3 are encoded by 2 genes DEFA1 and DEFA3 localized at chromosome 8. Location 8p23.1. DEFA1 and DEFA3 encode identical peptides except the conversion of the first amino acid from alanine in HNP1 to aspartic acid in HNP3. HNP2 is an N-terminally truncated isoform lacking the first amino acid. Human neutrophil peptides are found in human atherosclerotic arteries, inhibit LDL metabolism and fibrinolysis and promote Lp(a) binding. Human neutrophil-derived alpha-defensins, HNPs, are capable of enhancing phagocytosis by mouse macrophages. HNP1-3 have been reported to increase the production of tumor necrosis factor, TNF, and IL-1, while decreasing the production of IL-10 by monocytes. Increased levels of proinflammatory factors, e.g., IL-1, TNF, histamine and prostaglandin D2, and suppressed levels of IL-10 at the site of microbial infection are likely to amplify local inflammatory responses. This might be further reinforced by the capacity of some human and rabbit alpha-defensins to inhibit the production of immunosuppressive glucocorticoids by competing for the binding of adrenocorticotropic hormone to its receptor. Moreover, human alpha-defensins can enhance or suppress the activation of the classical pathway of complement in vitro by binding to solid phase or fluid phase complement C1q, respectively. The capacity of defensins to enhance phagocytosis, promote neutrophil recruitment, enhance the production of pro-inflammatory cytokines, suppress anti-inflammatory mediators and regulate complement activation, argues that defensins upregulate innate host inflammatory defenses against microbial invasion. In one small study, a significant increase in alpha-defensin levels was detected in T-cell lysates of schizophrenia patients. In discordant twin pairs, unaffected twins also had an increase, although not as high as that of their ill siblings. The virtual colony count antibacterial assay was originally developed to measure the activity of all 6 human alpha-defensins on the same microplate. Alpha-defensins in human plasma HNPs have been extensively studied as plasma marker of a range of diseases such as atherosclerosis, rheumatic diseases, infections, cancer, preeclampsia, and schizophrenia. Antibodies directed against fully processed HNP1 seem to have low affinity for the propeptides proins. A recent study used antibodies directed against proins to show that the predominant forms of alpha-defensins in plasma are in fact proins. Proins are exclusively synthesized by neutrophil precursors in the bone marrow and appear to be very specific markers of granulopoiesis. See also: defensin, alpha-defensin, beta-defensin, theta-defensin, cryptodan.